A very good evening to everyone. A very warm welcome to Dr. A.K. Sharma sir, all the participants, and our worthy resource persons for the day. Today is the second day of the 5-day faculty development program for our science educators from classes 6 to 10. It aims at enhancing teaching effectiveness by designing the resources around the latest theories and pedagogies. Such programs are integral to the continuous growth and success of teachers and has a positive impact on their students. The current era demands adaptive and innovative teaching methods. So without any more delays, let's start today's session with the blessings of Lord Ganesha, who is widely revered as the remover of obstacles and thought to bring good luck, the patron of arts and sciences, and the Deva of intellect and wisdom.
Jai Ganesh, Jai Ganesh, Jai Ganesh प्यारे जय गणेश जय गणेश प्यारे एक विशाल शोभित उज्ज्वल मोतियन की कंठमाल नैनार नारे जय गणेश प्यारे सुर ज्ञान राम तुलसीदास कनक नमन चरण में तिहारे जय गणेश प्यारे जय गणेश प्यारे
A very good evening. So we should start with the first session from our first resource person, Dr. Gurpreet Kaur, PGT Biology from Bharti Vidya Bhavan, SL Public School, Amritsar, with a total teaching experience of 20 years. She has been awarded the Best Teacher Award for Rotary Club, Amritsar 2023. She is a guide teacher for National Children's Science Congress for the past 20 years and has 5 research papers in a published form. So today, Dr. Gurpreet Kaur will give her session on the topic reproduction in plants. So over to you, ma'am, Dr. Gurpreet Kaur.
Thank you, Inu ma'am. Good afternoon to one and all present here. As we know, every ship needs a captain, and we all have Dr. A.K. Sharma ji with us. He's always there for us as a craftsman who will always try for our development training programs. As teachers' role never ends with their chapters are over. Today, I have been given an opportunity to discuss with you all on the topic of reproduction in plants. With due respect and permission of A.K. Sharma sir, sir, shall I start?
Please, ma'am, please.
Thank you, sir.
All the best.
Thank you, sir. So we all are here to study the topic of reproduction in plants. Words mean again. Production means to produce. As it is well said that reproduction is the basic necessity of life in every living organism. Because if reproduction is not there, species will become extinct. To maintain the species, whether it is animals, birds, humans, insects, any living organism I'm talking about, reproduction is a must. In general, we can say reproduction is a process of producing the offsprings. In general, we say offsprings, but technically we should say it as progeny that are biologically or genetically similar to the parent organism. Because reproduction helps in maintenance of species. Here we all have gathered to talk about reproduction in plants. So I'll be taking related to the plants only. But before starting, to study the importance of reproduction, I just want to say something. During COVID conditions, I was in a habit of watching the documentaries, and I was just watching a documentary of white rhinos. In that documentary, breaking news was appearing that white rhinos, there are only two left, one male and one female. But breeding was not possible in them because they were too old. And after 2-3 months, again, I watched the video in which it was said that male rhino is no more. Now, only two female rhinos are left. White rhinos, I'm talking about, my dear colleagues. Only two white rhinos are left. That two female rhinos, they are technically doing something so that they can be maintained. But here lies the importance of reproduction. That is, the species will not be maintained anymore. So we are talking about plants now. We cannot survive without plants. Plants are a boon to the whole mankind, and they provide us with food, medicines, fibers, everything we need for our survival. Plants need to reproduce continuously so that the plants can be maintained on the earth. Again, the importance of same reproduction as I was talking about, that it maintains the species. If species are maintained, balance of the ecosystem is whole and maintained over there. Now, talking about reproduction, the plants can reproduce asexually as well as sexually. When we talk about asexual methods, they can be leaves, they can be roots, they can be stems. So plants need to reproduce continuously to maintain the species, and sexual reproduction, the reproductive organ in plants, is the flower. If we talk about asexual reproduction, there are various methods: budding, fragmentation, vegetative propagation, spore formation, etc. But here, one thing I want to make clear, in plants only we talk about vegetative propagation. Why do we talk about vegetative propagation in plants? Because plants have the vegetative parts, that is roots, stems, and leaves. No vegetative parts are present in humans, in animals, any other living organisms. So plants are the organisms where vegetative propagation is very important, is very simple, is very cheap method. And flowers, they are not needed in asexual reproduction. Why so? Because flowers, they are the mode of sexual reproduction. And flower is, we can say, the sexual reproductive organ of the plant. So as asexual, A means without. So this means no two different sexes are required, no two gametes are required, and the plants produced by asexual reproduction will be the clones, will be the, as such, the same to their parent organisms. And one very important thing is that the plants produced by asexual reproduction thrive very well in the stable environments. Talking about sexual reproduction, that is by the flower itself. When we talk about sexual reproduction, we ask student, what is flower? That is the modified vegetative shoot. But before switching over to sexual reproduction, we should talk about asexual reproduction. Asexual reproduction is of two types. That is natural reproduction and artificial methods of reproduction. See, plants cannot do artificial methods on their own. If we are sometimes planting a cutting of a plant, a stem, they can propagate on their own. I'm using the word propagate. This means they are propagating through vegetative paths by natural methods only. So natural methods are budding, spore formation, fragmentation, etc. Natural methods, they include, I repeat once again, self-propagation of the vegetative parts. For example, the vegetative parts, they have buds. If we are talking about the small students, they have the outgrowths, they have the swellings. But when we talk about the technical word, that is, we should explain them that it is a protuberance. So we can say there is a bud, adventitious buds present, which lead to the formation of the new plant. So budding is the formation of asexual reproduction, where a new plant is developing from an outgrowth. That outgrowth is known as a bud. For example, plants such as ginger, onion, dahlia, potato, they grow from the buds. See, I have bought a potato. Is this visible to everyone? Is this visible to everyone?
Yes, ma'am.
Yes, ma'am.
So can you see the buds?
Yes, ma'am.
So these buds, these are the eyes. And the sweet potato, shakarkandi, I couldn't brought it. But the thing is, that is a root. This is a potato. Very simply, by showing in the class itself to the students, these are the buds. This means this is an underground stem and sweet potato is an underground root. Buds are appearing on them. Very simply, in a layman language, directly showing to the students, we can tell them the difference of underground stem as well as the underground root also. The buds are appearing. We call them in our own languages, eyes aa rahi hain, aankhein nikal rahi hain. So these are the eyes appearing. From where they are appearing? They are appearing from the nodes. Nodes kahan hai? They are on the stem. So that is, they are appearing. We will cut them, sow them, and plants of the potatoes can be obtained at home itself. This activity we performed in the school itself. We asked the students to bring the potatoes. The students were very happy to bring the potatoes with eyes propagating on them, with nodes propagating on them. And these nodes, these eyes, they cut with a part of the potato and sow in the soil. And they were very happy to see the propagated potatoes. Very simple activity by which they can have the new plants through the process of budding. So we can say in potato appears a small outgrowths, which are known as the buds, which commonly are referred to as eyes, and each of these eyes, along with the portion of the potato plant, they can be planted, which will grow up like the clone. We are using the word clone again over here, my dear friends, because they are exactly the carbon copy of the parents. No two parents are involved, no two different individuals are involved, so there will be no variations. No variations means they will be the carbon copy of the parents, so clones of the original potatoes will be obtained from this. So these are certain pictures of the sweet potato. Again, we can see the buds appearing from them. And here, a picture of bryophyllum plant is also given. This we can see the bud appearing. Next method is from the bryophyllum. That is from the margins of the leaves. We talked about vegetative propagation. That is, we talked about vegetative parts. That is the roots, stems, and leaves. So roots is propagated in asparagus. Can you see this plant? Can you see this, please?
Yes, ma'am.
This is an asparagus plant. If we plant this asparagus, a small asparagus plant in a small area, it will propagate over there and it will propagate to the whole area, whether it is a small pot, whether it is a soil substratum, it will cover the whole area only and only through propagation through the roots itself. Only once we have to plant that plant with the roots, rest whole area will be covered by a natural propagation through roots. This is an asparagus plant. This one is the bryophyllum plant. Can you see the leaves of the bryophyllum plant? Medicinal value, lots and lots of medicinal value itself. Can you see this? On the margins of the leaves, this is a very heavy leaf. And if the leaf touches the ground itself, only then also the plants can grow from here. And these are the margins of the leaves. Can you see the margins? The tapering margins. From here arises the buds. See, this is the broken leaf. Yahan pe ek bud thi. There was a bud over here. It has been broken. So from the margins of the leaves appear the buds, and these, when fall on the suitable substratum for the plants is the soil, the moistened soil, properly aerated soil. They fall on the suitable substratum. By chance, the lower leaf is there, it touches, then also the plants grow. See the nature, they are growing on their own, they are propagating on their own, they are maintaining their species, they are reproducing on their own. So this is what, again by leaves, so this is natural method of vegetative propagation. So three methods of vegetative propagation we have studied: through buds, through leaves, through stems, and through roots. That is buds, and the leaves, that is the bryophyllum plant. Next method is spore formation. Spores, wherever you people are sitting, wherever I am sitting, there are thousands of spores present around us. But you will also agree with this, we cannot see them because they are not visible with the naked eye. Just we can do one activity. What I did in my class, we kept a moistened bread in the class and we kept one rusk in the class, the dry rusk. What happened within 2-3 days? The moistened bread was full of fungus. There was no fungus on the dry rusk. Very simple activity, just to show the spores. Then we took that bread, moistened bread, which had fungus in the lab. We saw that fungus under the microscope, and it was clearly seen the hyphae, the mycelium, the sporangium with spores in it. So this is what I want to say, that spores are not visible with the naked eye. They are visible under the microscope, but thousands of spores are present around us. They're covered by a covering. That covering is known as cyst. That cyst or the covering, they make the spores dormant. Dormant means they can withstand unfavorable conditions such as high temperature, low humidity. They cannot germinate under those conditions. But under favorable conditions, these spores germinate and they grow into the new plants. Very simple example is of moss and the ferns. That is the pteridophytes. On the lower side of the leaves, they have spores present in them. We can propagate those plants through them also. But the rhizopus, that is the fungus, those spores are not visible. They can be visible only under the microscope. Very well tested when moisture conditions, that is favorable conditions, they are given to them. They can germinate very, very easily. The next type of asexual reproduction is fragmentation. This is also a mode of asexual reproduction. Fragments means pieces. If we cut the pieces of the plants, for example, in Spirogyra and Iggy, that each piece is a fragment of that. That is a small portion of the parent plant when cut into 2 pieces, 3 pieces, 4, 5, so and so on, so on. How many pieces it is being cut? Every piece, every part, or we can say every fragment will form a new organism of which is exactly like the parent organism. Because again, I'm saying it is an asexual reproduction, not the sexual reproduction. So we started about budding, spore formation, fragmentation. Now coming to the artificial methods of vegetative propagation, or we can say asexual reproduction. Plants cannot do cutting on their own. Plants cannot do layering on their own. Plants cannot do grafting on their own. So artificial methods include 3 methods, that is cutting, layering, and grafting. Cutting is from the stems. That is best done in case of rose plants. But the problem is unknown people cut, just a direct cut is given on the stem, but we should give a blunt cut, but we can say a V-shaped cut, or we can say a oblique cut. Why an oblique cut is given? To increase the surface area. That is when planted in the soil, that should be properly aerated, that should be properly watered regularly. In dry soils, the propagation is not easy. So by cutting, any plant can be propagated very, very easily. Second is layering. Hibiscus can also be propagated by cutting. Bougainvillea can also be propagated by cutting. Second one is layering. See, in layering, what we have to do is, we have to make a layer on the stem surface. I'm taking a simple plant of asparagus. Here, the layering do not occur, but the layering occur in jasmine. Why I have taken the stem? Just to show that this is a weak stem. That is, it can be folded. A U-shaped bend can be given in this. This is a stem. This is of hibiscus, not of guava plant. I tried to give a bend in this. You can see the wound over here. Can you see the wound over here? Can you see the wound over here?
Yes.
Yeah. If it is bent, see, it will again break. It will break. The bend cannot be given to this stem. So layering is carried out in different plants which have weak stem, that is jasmine plant. Supposing here is the soil, here is the plant growing. We just give a U-shaped bend. We will bend this stem, give a U-shaped bend, cover it with the soil, and this is known as soil layering or the mound layering. Because it is very near to the soil, very soft stem, weak stem, so it can easily be given a U-shaped bend. From that U-shaped bend, after some time, roots will appear and the new plants can be propagated. But in case of, we can say, the air layering, such as in case of guavas, what happened? We just take out the bark of the guava plant because it is above the ground surface. It is not near the ground surface, but we do not cut the xylem and phloem. Then we cover it with the moist soil having various hay particles and other things in it. We just give supposing this is a stem we are taking, we just cover it with the moist soil, and this part of the part which is covering it, this shape, this is known as gooty, Gootte, this is known as gooty. Then we cover it with a polythene so that there is no exchange of heat and matter, we can say, so that the soil do not become dry. After some days, we can see the roots appearing from it. We can cut that plant and can plant it anywhere. As it is in the air, as it is in the atmosphere, this is also known as air layering. So, my dear friends, we should keep in mind in the soil layering, the stem is weak, but in case of air layering, it can be done in the hard stems, or we can say the trees. This is a method of grafting. In this, two things are taken. One is stalk, one is scion. See, supposing there are two varieties of mangoes. One variety is resistant to diseases. Resistant to diseases means its root portion is resistant. One variety is a very good high yielding variety. For example, nowadays new varieties of mangoes are there which can give mangoes or can give the yield after 1 year. Earlier, when we used to propagate mangoes, they used to give yield after 5 years. Let it be the same thing. One variety is having disease resistant roots. It bears mangoes after 5 years. The other variety is a very good variety, very tasty mangoes, high yield. But the problem is it is not disease resistant. If it is not disease resistant, then if the roots are susceptible to diseases, the whole plant will die. But what we want is we should have the characters of both. What we can do is we can have the root portion of the wild variety, that is, which is disease resistant, and we can have the shoot portion of the second variety, which is high yielding variety. How can it be done? We just take a stalk, that is the root portion, which is disease resistant, and give a wedge shaped cut, or T-shaped grafting can be done, or any type of grafting can be done. Then we take a scion, that is the shoot portion, which is high yielding variety portion. This stalk, that is disease resistant, and scion, that is high yielding variety, they both are tied with each other. The most important thing in this is that is the cambium should be intact. Cambium should be intact means the xylem and phloem should be intact. Why? Because the absorption part, the movement, the translocation should be there. After some time, we see the grafted portion and a new plant arising, which has both the characters, that is, it is disease resistant also, as well as it is high yielding variety also. Let's see a slide video which will be telling us about the technique of grafting. I think the video is not being shown. Just give us a minute, please. There must be some technical issues. Sorry. So we will continue further. Another is the tissue culture. Another method we can say is the tissue culture. So tissue culture is a technique, we can say, of micropropagation, where the plant cells or the organs are grown in a nutrient medium, where the micropropagation is the production of thousands of identical plants. We see tissue culture. See, I'll give a very simple example. Mint, pudina, that is there in our homes. It always comes with stem and leaves. Roots are not there. And if we try to propagate it through stem, it will never be propagated. But if we take just a small tissue of it, small tissue, maybe nodes, and we plant it, I should not use the word, we should propagate in the culture medium by giving everything to them from that tissue only, that is from the cells when they break down into callus. Tissue break down into callus, that is the cells. There is a very important property of cellular totipotency. Cellular totipotency is a property in which every cell has the ability to grow into a complete new individual. So the tissue culture is a technique in which a small tissue is taken, that is the explant is taken, that is put in a medium in a test tubes, and that is propagated under lab conditions. So first of all, the simple way is to select the mother plant. What desired quality plant we want to propagate. Its multiplication is done, rooting and acclimatization is done, and transferring is done to the new plant. See, a small plant is to be acclimatized. Acclimatized means we have to train it to adapt to the harsher conditions. A small baby is there, he or she cannot do anything. We train it. We train him slowly and slowly. Similarly, we are growing a plant from a small tissue. Cells, they are propagating into a complete new plant. So what is happening? That roots will appear, shoots will appear, because this medium, we are providing them the complete medium. That medium is being provided to them, which is the plants propagated in medium. They are to be acclimatized. First of all, they will be grown in the sands, then to the soils, and then to the harsh conditions of the crop fields. So this is how the micropropagation is done of the plants. From various parts, the micropropagation can be done. The first one is the meristem culture. Meristems, as we know, root apical meristem, shoot apical meristem. That is the meristematic cells which are on the verge of division. The cells which divide, they are the meristematic cells. So cells which are on the verge of division, that is the meristematic cells, if we take that tip, the primordia, the apical meristem or the shoot apical meristem or root apical meristem, they can propagate on their own in the plants, in the test tubes, and then acclimatized so that they can be transferred into the soil. So by this method only, disease-free plants can be produced. They can be successfully used for rapid multiplication of various plants. I want to quote an example of orchids. Orchids were very difficult to propagate, but through tissue culture and micropropagation techniques, they are very easily propagated in the biotechnological labs nowadays. Another methods are callus culture, suspension culture, embryo culture. Callus culture, the method is that is the selected plant tissue is placed in artificial growing medium until the callus is formed. What is callus? Callus is the cells from the explant taken. That is, they are transferred into a culture medium containing plant growth regulators for the induction of adventitious organs. After a few weeks, new plantlet is exposed gradually to the environmental conditions. My dear friends, here again, the new plant exposed gradually to the environmental conditions means that is to be acclimatized. This means from the test tubes, first of all, they will be grown into the pots containing the quartz sand, then to the soil, then to the crop fields. Suspension culture means we are growing the plants in liquid medium. That is, the cells or the groups of the plants, they are dispersed and allowed to grow in an aerated or sterile, we can say the liquid, which is the culture liquid. This culture liquid we use is most probably the MS medium. That is Mirashige and Skoog mediums. They contain all the antibiotics, they contain the sugars, they contain the auxins, the cytokinins. That is, we are giving them a well-prepared medium in which the plants can grow. Next is the embryo culture. Here the plants, they are directly grown from the embryo when placed in culture medium with proper nutrient in an aseptic situation. Next slide, please. Now coming to the flower. These all are the methods of asexual reproduction. Before coming to the flower, I just want to show you one thing. We talked about ginger. Is it visible? Which one is ginger? Can you please tell me which one is ginger? This one or this one? My dear colleagues, this one. Okay, what is this?
Both are appearing
Ginger?
same.
It's ginger.
And this one? Again ginger?
Turmeric.
Turmeric.
This is turmeric. Yes, the students don't know much about it, but I have cut the turmeric over here. So we can show these things to the students in the class because they can be propagated vegetatively through asexual methods. So these are a must because we go on teaching and teaching. We just don't show them the things what they are being taught. So please, whatsoever the layering, the grafting we are teaching them, just tell them about the plants. Just take them to the nature and let them propagate the things. They will be very happy. Now coming to the flower, that is the reproductive structure. When we go to the class and ask them about what is flower, they just say the showy portion, the flowery, the nectar containing part, the aerated, we can say having scent. This is flower. But what is flower is? That is, flower is the reproductive organ of a plant. See, plants, they are growing. Seed is there. We sow the seed. It becomes a seedling, the sprouts we can say. From the seedling, it becomes a plantlet. Plantlet becomes a plant. Vegetative growth of the plant is there. Then comes the flowering and then comes the fruiting. So what is there in this is that flower is the reproductive organ because it contains both male and the female gametes in it. Male gametes also in the flowers, female gametes also in the flowers. So flower is the part which is great importance to humans. It has aesthetic, ornamental, social, cultural, and religious values. They are structurally divided into following parts. Number one is calyx and corolla. Calyx, the children know it as sepals, the green colored sepals, which protect the flower during the bud condition. Every one of us know the bud. This is in this shape. And this is totally covered by green colored leaves, the students say, but they are the sepals. They're protecting the bud during the flower during the bud condition. Next one is the corolla, which is very brightly colored. It is having fragrance also. Why this is having fragrance? Because it had to attract insects for pollination. Pollination is a must for reproduction, otherwise reproduction will not take place. Third one is the androecium and the fourth is the gynoecium. These are the whorls in a flower. Androecium, they are the male reproductive parts of a flower, which consist of stamen having anther and filament, and the gynoecium, they are the female reproductive part of the plant, which consist of the pistil or the carpel having three parts, that is stigma, style, and ovary. My dear friends, let us watch a video of this dissection of a flower. We are trying it from the link due to the technical issues. Just a minute. Wait a minute, please. Can you see this, please?
Yes, we can see.
Is it visible?
Mm. Yeah,
Yes,
it is
ma'am.
removing the petals from a flower.
I've shared the link in the chat box. If you want to see, you can see it from there also. So coming back to the flowers, from the link I've shared, that is for the dissection of hibiscus flower, we have come to know about that is the flowers may be unisexual also, the flowers may be bisexual also. Unisexual flowers means it may be either the male flowers or it may be either the female flowers. This means the whorls, that is the sepals and petals, will be there. These are accessory whorls. But the whorls, that is the staminate containing the male whorl or female whorl, that is the pistillate whorl, only one whorl will be there, not both the whorls. Unisexual, single sex will be present, either the male or the female. If it is having male part, so it will be a staminate part. If it is having a female part, so it will be a pistillate part. This means they will be unisexual flowers. But unisexual flowers, we know it is further monoecious or dioecious. See, in papaya plant, I heard someone saying, हमारे घर में पपीता लगा है, उस पे plants में flowers, fruits नहीं होते. Why so? Why so, my dear friends?
Because it is unisexual
It is a male plant.
male plant.
Only the female plants can have the fruits. Only they can bear the fruits. But condition is if the pollen they are coming from the male plant sidewise. Kahin pe male plant is there, only then the pollen can come. We see the cucurbits, ghia, karele and all, they all have the male flowers as well as the female flowers on one plant only, though they are separate. So they are monoecious. Mono means single, dioecious, di means two. So monoecious and dioecious plants. So they both are unisexual flowers. Bisexual flowers are the flowers which contain both male and the female reproductive organs in the same flower. As we saw in the dissection of the hibiscus flowers, the male and the female reproductive organs, they were present in the same flower. So these are the bisexual flowers. Next is the reproductive structure of a flower. Because we are talking about the male and the female parts, so we should be very much aware of the androecium and the gynoecium, that is the stamen and the pistil. The androecium and gynoecium consist of male reproductive parts and the female reproductive parts. Androecium has anthers and filaments. When we do the dissection, when we see the yellow colored pollens, that is the anthers have pollen grains in them. In this diagram, very clearly it is visible in the pollen grain is a sac-like structure that contains microsporangia. That is a sac-like structure. Microsporangia will contain pollens, that is the microspores. But one thing we should make the students aware is about the ploidy. Though some say it is a topic of +2 or the higher classes, but I think the students should know because we teach them the concept of XXXY. There we talk about 46 chromosomes in male, 46 chromosomes in female, 23 each, one each from 23 from father, 23 from mother, only then the ploidy can be maintained. Because if one chromosome or the other is less or more, ploidy changes, it changes the complement of the chromosomes also. So here also they should be very well aware that pollen grains or the microspores, they are haploid. Similarly, the egg in the, when the male gametes will be produced in the haploid, they will be haploid. Similarly, the female gamete, that is the egg, should be haploid, so that on fusion, when zygote is formed, that should be diploid. That's why a little bit of information should be provided to the students regarding the microsporangia. Very simple, because it will contain spores. Which spores? Microspores. What are that? The pollen grains. What are that? The male reproductive parts. Similarly, the filaments, they are the thread-like structures that are attached to the anther, and they keep the anther in place. Because otherwise, they are just hovering here and there. So filaments, they keep the anther in place. Then there's the microsporangia contain the spores, and the topic of microsporogenesis should be told to them, that is the meiotic division. They read mitosis and meiosis in 9th class, but it should be explained to them that the microsporangia contain microspores after meiosis, that is from 4 pollen grains, or we can say the microspores, the haploid they are formed, so that the ploidy in the individual plant can be maintained. This is the structure of gynoecium, or we can say the female reproductive part. It is the female reproductive part, contains stigma, style, and ovary. Ovary contain the ovules, which are present inside the ovary, which contain the female reproductive gamete, that is known as the egg. So pistil has 3 parts, we can say stigma, style, and ovary. Stigma is on the top of the carpel. Here we should tell them about the chemotropism also, because we are going to talk about pollen pistil interaction. Pollen pistil interaction means that is the stigma. That is, the stigma contains the chemicals which can check whether the pollen they are of same species or not. If the pollen they are of the same species, they can be given entry into the ovule to reach the egg to fertilize the egg. If they are not of the same species, they are discarded and they are no more viable. Chemotropism is very important. In general, in a layman language, we can say that there is a talk between them. So the pollen of the same species is given the entry, otherwise pollen they are discarded. So stigma contains the chemicals and check whether the pollen which is landing on them is of same species or not. Similarly, as we talked about microsporangia, there is megasporangia. Megaspores are the females and microspore are the male parts. So megasporangia and the megasporogenesis. Genesis means synthesis of megaspores. Where will they be present? In a megasporangia. So megasporangia will contain megaspores, which will have the egg, and it gets fertilized, we can say, with the male gametes, which are 2 in number. Here, one thing I want to say is that whatsoever is present over here, we can tell them about this ovules, the placenta, the seeds through a pea. The students, they can be given peas to take out the seeds from them very easily. They can be shown the placenta in it. So before fertilization, before the male gamete reaches the female gamete, the process of pollination takes place. One example, one true thing I want to quote over here. During COVID condition, there was a news that in China, honeybees were not there. And for the pollination of pears, the persons were standing on the trees and brushing the pollens on the flowers so that pollination can take place and pears can be obtained. So pollination is a very, very important step for further fertilization of the male and the female gamete. See, the life starts from embryo. What is life? Just an embryo. The male gamete will fuse with the female gamete. It will form zygote. Zygote is just a cellular mass. It will go on dividing and dividing will form embryo. Embryo will differentiate. Whether it is embryo of plants, it will differentiate into plants. Whether it is embryo of humans, it will differentiate into humans. Bird into bird, and so on, so on, so on. So pollination is a must. It is an agency. It is a process to transfer the pollen from to the stigma of the flower. So it is further of two types: self-pollination and cross-pollination. Self-pollination means either from the one flower to the same flower of the same plant, or one flower to the other flower on the same plant. So if it is on the same flower, this is autogamy, auto, similar, same. And if it is on the different plant but same flower, it is geitonogamy. And then second one is the cross pollination. This is also known as xenogamy. That is, the pollen grains, they are transferred from anther of a flower to the stigma of another flower or another plant. So this is what cross pollination. My dear colleagues, cross pollination always brings variation, because variation leads to survival, variation leads to evolution. That is, they can survive in the adverse of the conditions. So self pollination, plants have various devices for preventing self pollination and going from cross pollination, but certain plants are there where cross pollination is not possible, so they undergo self pollination only. So we can try this video. Otherwise, I will share this video in the group. I'll try to show you from my phone only. Otherwise, we will share this link in the group. Is it visible and audible? Okay, I'll show you. Is it visible?
Shoot away. Let it do its job. It's only trying to help the plant pollinate. Well, interesting question. Hey friends, I'm sure you must have heard of it.
Is it visible?
Let us explore the reproductive world of
Yes, ma'am.
plants.
Yes, ma'am, it
Answer
is visible.
an evergreen question. What is pollination? Zoom in. So, what is pollination? It is the process through which plants reproduce, meaning birth, fruits, and
Let us
But the vital question is, how does it happen? To know that, first we need to talk about the reproductive parts of the plant. And what are they? Well, are consists of both male and female reproductive parts. The male reproductive part of the flower is called a stamen. It is made up of anther and filament. The anther has a sac-like structure that contains pollen grains, which are the yellow powdery tiny particles that play a crucial role in reproduction. And below the anther is a long stem-like part called the filament.
let
On
us
the
know
other hand,
about the
the
process of
part of the
pollination.
flower is called pistil, consisting of stigma and ovaries. The stigma has a glue-like substance, whereas the style is a thin stem-like structure that connects the stigma to the ovary, a sac-like structure that contains eggs which reside in ovules. Now that we have learned about a plant's reproductive parts, let us know about the process of pollination. Pollination is a process through which pollen grains are picked from an anther and transferred to a stigma. This process can occur in two ways: self-pollination and cross-pollination. So, let us look at them in detail. Self-pollination occurs when the pollen from the anther is deposited on the stigma of the same flower or another flower on the same plant through wind and water. whereas cross-pollination occurs between different plants in which pollen grains are transferred from anther of one flower or plant to the stigma of another to each bird, wind, etc. known as pollinators. For example, when a bee sits on a flower for sipping nectar, the pollen gets stuck on its body, and when it flies to the next flower for more nectar, the pollen from the bee's body gets deposited on the female reproductive part. And when that happens, the pollen gets slipped into the ovary and enters the ovule, where seeds are formed through a method called fertilization.
So here we could see the process of pollination, that is self-pollination, autogamy and geitonogamy, and cross-pollination, that is xenogamy. So there are various agents of pollination, that is abiotic and biotic. Abiotic means the non-living ones, that is the air and the water. And biotic means the living one. They can be humans, they can be animals, they can be bats, they can be birds, insects, anyone working in the fields or having nectar or any other activities with the flowers. So they have different characteristic features. Abiotic, obviously they will be light. They are to be carried over to long distances. No agency as such is there, because as the water is moving, they are moving. As the wind is moving, they are moving. So they are to be produced in large numbers. They need not to produce any nectar. They should not be fragrance. They should not be colorful, but they should be having some structures which are carrying them to long distances. That should be plentiful in number. This is the slide showing various living agencies, the biotic agencies, that is insects, that is entomophily, ornithophily, that is bird pollination, chirotelophily, that is by bats, and melecophily, that is by snails. Now coming to the pollen-pistil interactions. Pollen-pistil interactions, as I was talking about in a layman language, we can say it is a talk between the pollen and the stigma. Very easily we can tell students as if we are teaching them about hydrotropism, phototropism, geotropism, and the most important thing is the chemotropism, that is the chemicals present on the stigma which will recognize whether the pollens they are of the same species or not. If they are of the same species, they will enter the ovary, reach the ovule, and will fertilize the egg. If they're not of the same species, they will be discarded and they will be no more viable. So my dear friends, the sexual reproduction in flowering plants is also known as double fertilization. This is also known as triple fusion. Some teachers, they say we should not tell the students in details about how many cells are there, how many haploid are there, how many diploid are there, but I'm of a view we should tell them. How can we explain double fertilization? How can we tell about triple fusion? How many cells they are fusing? That's why 3 cells are fusing. That's why this is triple fusion. But if we are not going to tell about them about the cells, different type of the cells present in the ovule, how can we tell about what do you mean by double 2 fertilizations taking place? So we have to tell them about the meiosis, the 8 cells produced, how they are produced, 4 towards micropyle, 4 towards chalaza end. Now, what is micropyle? What is chalaza? Micropyle is a small opening between the 2 layers, 2 coverings, 2 outer protective layers of the ovule. Those protective layers or the coverings, they are the integuments, which will later become the coverings of the seed, that is testa and tegmen. In the micropyle, the ovule to the ovule will reach the 2 male gametes, which are released to the pollen grains. One of the male gamete will fuse with the egg, which is towards micropylar end. One of the male gametes will fuse with the 2 polar nuclei, which are coming in the center from the 2 ends of the ovule. Those 2 polar nuclei, now they are fusing with the male gamete. 3 cells are fusing, that will be triploid in nature, and the process is triple fusion. The zygote is formed by fusion of male and female gamete. So this is what we can say, that is the male and the female gamete fuse to form the zygote. This zygote is diploid in nature. So ultimately, this will form seed. What seed is? it is having embryo and endosperm. Am I audible?
Yes, ma'am.
Okay, thank you. Thank
Yes,
you.
ma'am. Yes,
Thank you.
ma'am.
Thank you. Thank you. Thank you so much. So what is there in this is, I was talking about zygote leading to formation of embryo and primary endosperm nucleus leading to formation of endosperm. Dear friends, we should tell them, just bring a rice grain. Rice grain means just a seed or a grain of rice, and show them towards the light. When you are going to show them towards light, one will be whiter portion, one will be translucent. It contains embryo and endosperm. Very easy to teach them where embryo is, where endosperm is, because it is clearly visible in that. Very easy to tell them. Just bring rice grain from homes, show them the rice grains. One will side will have embryo, the other will have endosperm. So moving further, again, a video is there if it is working. Otherwise, I'll show you from my phone itself. This is related to the fertilization process. I'm sorry for this inconvenience, but technical issues, we cannot say anything.
Don't say that. Gold are the reason we get fruits and vegetables, and their creation is no less than a wonder. Hey friends, in today's episode, let us learn about the formation of seeds and answer a life-proving question. What is fertilization? Zoom in. Plants. They are nature's best gift to us as they provide food, clothes, and even shelter, because of which they are known as universal or primary producers. And as we know, just like other living things, plants have a life too. And just like us, they tend to respire, develop, excrete, and also reproduce. Yes, my dear friends, plants majorly reproduce through three main steps, which are pollination, fertilization, and lastly, germination. We have already learned about pollination and germination in our previous videos, which leaves us with the process of fertilization. And what is that? Fertilization can be defined as a process of sexual reproduction in plants, in which the male gametes or pollen merge with the female gametes or ovum to form a diploid zygote, or the eggs that later develop into seeds. But the vital question is, how? Well, it all starts with flowers, which are the reproductive organs of plants. Yes, as we know, during the pollination process, the pollen gets transferred to the female reproductive organs and slips inside the ovary. After which, the pollen tube opens into the ovule and bursts into the embryo sac. Here, the male nucleus unites with the egg's nucleus inside the ovule, where a zygote is formed. And how does this happen? For that, let us look inside the pollen first. Pollen grains are yellow, powdery substances that consist of two layers.
So this is the video related to, we can say, the germination, that is the fertilization.
It travels all the way
This
down
is how
to
it is
the
moving.
microphile, where one of the gametes joins the egg and forms a diploid zygote, whereas the other gamete joins the two polar nuclei to form a triploid endosperm nucleus, which will transform into a nutritive tissue and provide nourishment to the developing embryo. This process is called double fertilization. Every flowering plant undergoes a double fertilization process at least once, after which the ovary transforms into fruit.
So my dear friends, was the video related to double fertilization. Sorry for the technical issues. I will share all these links in the chat box. If you want to watch, you can have. So moving further with the slides, that is, we were talking about double fertilization. So these are the fertilization events which we had seen in the videos. That is, the one of the male gamete fuses with the polar nuclei leading to triple fusion. The other fuses with the egg leading to the formation of zygote. Zygote forms the embryo that will be diploid, and the polar nuclei on fusing with the male gamete forms the endosperm, which will be triploid in nature. So these are the post-fertilization structures, the answer to the fertilization, formation of endosperm and formation of embryo. Endosperm formation means that is, it is leading to the formation of a nutritive tissue. See, seeds are germinating in the soil. They also need nutrition. How can they germinate in a dry soil, which is totally without aeration, without water, and without nutrition? We also need nutrition to grow. Development is also required. This means some nutritive tissue. See, nature has provided wonders to them. So the nutritive tissue present in the seeds is the endosperm. So endosperm will provide the nutrition to the growing embryo. And embryo, the life, the starter of life, the starting kit for the future life, will lead to the formation of roots and shoots. हम लोग घर में करवाते हैं, हम लोग sprouts बनवाते हैं. That is the embryogen. That is, we can say, the seedling is being formed. So this is the formation of seed and fruit. Seeds, this is a monocot seed and this is a dicot seed. Seed, we eat maize, corns, that is a monocot seed. We eat kidney beans, they are the dicot seeds, 2 cotyledons. And this is a fruit which has the coverings, that is the epicarp, mesocarp, endocarp, this is the pericarp. And inside the seed will be present the embryo and the endosperm. See, some students, they bring mango seeds to school, but they bring it with endocarp. They don't, we can say, distinguish between what is endocarp and what is seed. So we planted mangoes in the school. One we planted with endocarp, one we planted after cutting the endocarp. The cut endocarp seedling that grows very well in time, while which had endocarp that took some time to grow, thus showing that this is the covering of what? This is the ovary wall, the third part, that is the endocarp, and the seed is, that is the ripened ovule, is present in between. So such small activities, when performed by the students, they are very happy to perform and they can grow plants on their own. Next, we can say this is the summarized form of sexual reproduction. That is, the life started from enthesis, pollination, pollen tube germination, that is going to the embryo sac, zygote, embryo, seed, germination, again a sporophytic plant, again the same steps. That is reproducing, producing again and again. So this is the summarized form of sexual reproduction. One thing I just want to share with you is the artificial hybridization. That is, asexual reproduction is taking place, vegetative propagation is taking place, sexual reproduction is taking place. But what we want to have new things, because the humans, they have the desire to have the best quality of the things, the food, the plants, everything should be the best. So that can be attained by artificial hybridization. That means distant hybridization by breeding methods. That can be done by emasculation, bagging, tagging, and rebag. Emasculation means removal of anther from the bisexual flower. Then the bagging is done. Bagging means that is, we are doing covering the removed anther flower. This means that is having only and only the stigma. Why we are doing bagging is because if it is not covered, if the bag is not put over it, then what will be there? Any pollen can do the cross-pollination over there. So it's better before opening of the flower, bagging should be done. So we can say emasculation should be done so that we can say after doing emasculation, covering is there. That is known as bagging. When bag is being put, we put the tag over there so that the tagging is done, where it is written, at what time it was done, why it was done, on which day it was done, emasculation is being done, what we have to do further. Then we pollinate, that is dust the pollens over it on our own. How do we dust? We can do it with the help of a brush, or we can do it with the help of a any other directly from the pollens from the plants. This we do is of our own desired plant. That is, we can say the own desired quality. After that, immediately rebagging is done. Why rebagging is done? So that any other pollen do not come and interfere with it. I again had a short video. I will send the link in the chat box if it is not working. So I'll try to open it. I'll try to play it in the phone itself. If it is not working, then I will send that video in the cell that we can say the link in the chat box. I'll send the link in the chat box so that you can see the wonderful video of emasculation, tagging, bagging in the chat box. In short, we can say the steps involved in hybridization are selection of parents, evaluation of parents, because we need to have the parents which variety we want to have. Then is the emasculation, that is removing of anthers. Then after removing of anthers, bagging is being done. See, you can see in the diagram here, in the pic here, the bags are being put over the plants where emasculation has been done. When I was young, our neighbors were there in the university. What they used to do, they had cucurbita plants in their home, ghiye ke, but they used to dust something of the small creamish color flowers. I used to ask them, what are you doing? So they used to say, that is not having pollination property over here. So we are dusting the pollens. See, knowingly, unknowingly, I remember today, they were doing pollination. They were doing hybridization. But that is not a case of artificial hybridization. We should say they were doing pollination. But in the same way, we can do hybridization, artificial hybridization. But the thing is under very controlled conditions by emasculation, bagging, tagging, pollination, and then rebagging. Whatsoever the plants produced will be of our own desired will. So to conclude my topic today, we can say reproduction is very vital because it maintains the species. Without reproduction, a very small word is there, extinct, but a very big thing to be considered. Extinct means that it is not there now. So reproduction is very vital to ensure variation and perpetuation in organisms. Reproduction is a very crucial step in the lifespan of organisms, which ensures their perpetuation as a distinct species. The success of the process is largely influenced by two components: inherent biology of organisms and interactive ecological domain in which they grow and flourish. A very important thing, because the vegetatively or asexually reproducing plants can grow in favorable conditions in which they are growing, but others which are having variations, they can grow in distant environments. That is, they can grow and adapt to the changing environmental conditions. So we can say plants optimize reproductive success through interaction between these components of reproductive structures and ecological factors. One thing more I want to share, my dear friends, this is a video I'll share. This is a video of a homework being given to the students. You can give a very beautiful project. The students can make it as a herbarium by planting, taking the plants, by taking the parts of the plant, pasting them, and about the sexual reproduction, they can give the details. That's all for today. Thank you so much. If you want to ask anything, please. Thank you, dear friends. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
My sincere thanks to Dr. Gurpreet Kaur for her exceptional presentation. Ma'am, your insights and expertise has made a significant impact on all of us. And I'm sure that all my participants would agree with me.
Thank you, ma'am. But sorry for the glitch. I'll share all the links in the chat box. Thank you.
So now I invite our next resource person for the day. Mr. Hartinder Kumar. PGT Physics from Bharati Vidya Bhavan Public School, Amritsar, with a total teaching experience of 27 years. He has been awarded the Bhavan Jindal Award for the Best PGT 2012 and CBSE Best PGT Physics Teacher 2018. He is a CBSE resource person. He is a guide teacher for National Children's Science Congress, CBSE Science Fair, Inspire Award, National Innovation Foundation for the past 27 years. So over to you, Hatinder sir. A very warm welcome to you.
Thank you,
Thank you, Inu ma'am.
Thank you, sir.
Good evening, everyone. Namaskar. I welcome you all to the Faculty Development Program for the Science Educator, organized by Section Bharati, Bharatiya Vidya Bhavan, Delhi Kendra. My sincerest gratitude to Dr. A.K. Sharma sir for providing me this opportunity to interact with the science educators across Pan India, where we can discuss that what are the latest pedagogies which we can use in the class to meet the vision of National Education Policy 2020. With the permission of respected Dr. A.K. Sharma sir, I'm going to start my session on the topic magnetic field produced by current carrying conductors for the class 10. So today's session is based on magnetic field due to current carrying conductor. So we all will share our views on this topic. What are the different pedagogies which we can use to make the concept of this topic clear to our learners? And we will also discuss the various type of activities which we can perform to make the concept clear. My dear friends, the Ministry of Education has introduced National Education Policy 2020. And as per the instructions of CBSE and Ministry of Education, all learning instructions, all teaching learning processes should be aligned with the vision of NEP 2020, which says to provide an education to all means to all, which is a blend of 21st century skills and great Indian ethos. My dear friends, the world is changing every second. In the coming 5 years, the new professions will come, whereas the older ones are disappearing. The technology is changing every moment. And if we want that our children should survive in the next 5 to 10 years, then they must possess the 21st century skills. Not only 21st century skills, but the great Indian ethos also. They must have a strong value system. The Ministry of Education has identified 3 skills as a 21st century skills. And they are known as learning skills, literacy skills, and life skills, as you can see on the screen. There are 4 learning skills. They are named as 4 C's: the critical thinking, creativity, collaboration, and communication. There are 3 literacy skills identified by CBSE, Ministry of Education. They're known as IMT, the information literacy, media literacy, and technology literacy. And the most important, the life skills. They're known as FLIPS. There are 5 life skills identified by Ministry of Education: flexibility and adaptability, leadership and responsibility, initiative and self-direction, productivity, and social and cross-cultural interaction. My dear teachers, my dear friends, these 21st century skills are very, very important to develop among the learners. And we should plan our lesson every day to inculcate, to develop the value system as well as the 21st century skills among the children. To achieve the vision of National Education Policy 2020 and to achieve the desired learning outcomes, the CBSE has defined certain curriculum objectives. The first one is the teaching learning process should aim for 360-degree holistic development. The role of teacher is not to provide the information to the children. Our role is not limited to provide the knowledge to the children. The aim of teaching learning process should be 360-degree holistic development. That includes 21st century skills along with the great Indian value system. The children in the classroom and outside the classroom must be given enough opportunities to construct the knowledge rather than rote memorization. We should discourage the rote memorization in classroom as well as outside the classroom. The children must be allowed to construct the knowledge. The children must acquire essential skills and competencies to develop problem-solving skill and decision-making skill to solve the real-life problems. My dear friends, as respected Dr. A.K. Sharma sir said yesterday, that we are to make our children resilient, make them resilient. If somehow they face failure in their life, they must be capable of standing again. To make children resilient should be the aim of education. Along with it, they must have a decision-making skill so that they can take the right decision in their life. They must have a problem-solving skill to solve the real life problems. Imparting knowledge is just a drop in the ocean in an education system. The teaching learning process include innovative pedagogies such as experiential learning, art integrated learning, toy-based learning, storytelling, gamification, ICT integrations. So these are the different innovative pedagogies which are suggested by CBSE to all schools across Pan India. And we, the teachers, have responsibility. We have duty to follow the instructions of CBSE. We are to inculcate these innovative pedagogies in day-to-day teaching learning process. Only then we will be able to achieve the vision of NEP 2020. My next slide is on syllabus versus curriculum. We all teachers in the beginning of session always look on the syllabus. That is the topics to be finished across the session. We hardly look at the curriculum. Syllabus, as I said earlier, it's just a drop in the ocean. The curriculum is ocean. The curriculum is an holistic approach towards obtaining desired learning outcomes with the help of conducting various n number of activities within the classroom as well as outside the classroom. To finish the syllabus is not the objective of education. We are to perform the various type of activities within the classroom as well as outside the classroom. And this is the only way we can prepare our future citizens. This is the question paper design as issued by CBSE. My dear friends, if you look at this design, you'll find that about 68% of the questions will be asked from low order thinking skills. What are low order thinking skills? I will be discussing with you the Bloom's taxonomy. 68% questions will be there to test their low order thinking skills, whereas 32% of the questions will test their higher order thinking skills, which are known as competency-based questions. So we are to provide instructions to our children. We are to decide our learning outcomes, learning objectives, all assessments, formative as well as summative, in such a way that can develop low order thinking skills as well as higher order thinking skills among children. So every learner in the classroom should have ability to respond to the questions which are based on loads as well as HOTS. So we need to understand first the question paper design before deciding or before planning a lesson in the classroom. My dear friends, to achieve the vision of NEP 2020 and to develop the required competencies as described in the question paper design, the key tool is, the significant tool is Bloom's taxonomy. So before planning any assessment, any lesson, we need to understand the Bloom's taxonomy. My dear friends, all question papers at all the levels, from school to CBSE and to JEE NEET, all levels are designed as per the Bloom's taxonomy. Here on the screen, you can see the 6 cognitive levels of Bloom's taxonomy, from simple to complex, from concrete to abstract. These 6 levels of cognitive domain, that is the thinking skill, will help all the educators to decide their learning objectives, their learning outcomes, their question paper design. And if we follow this Bloom's taxonomy 6 levels while planning a lesson, while planning any assessment, our children will do wonderfully well. And in that way only we will be able to achieve the vision of NEP 2020. The simplest level in Bloom's taxonomy, that is the cognitive domain, the thinking skill, is the knowledge, where the questions are based just to recall the informations, definitions, the units. And then comes understanding, that is the comprehension. So when we ask from our children, describe this, explain this, we are actually asking the questions based on understanding. And next is the application, where the children are able to apply the knowledge they have learned in the classroom on unknown situations. Draw a diagram, solve the numerical problem. These questions belong to the application level. Knowledge, comprehension, or understanding and application. These 3 levels are called as low order thinking skills. 68% of the questions in 10th standard board examination will be based on these 3 thinking levels. That is knowledge, understanding or comprehension, and application. And then comes higher order thinking skills, analysis, evaluation, and creation. The remaining 32% questions will be based on analysis, evaluation, and creation ability of the learners. In analysis, what we do? We split a concept in the micro parts. The splitting of a concept into micro parts is analysis. Write the properties of metals, write the properties of a bar magnet, write the properties of magnetic lines. So these are the questions which are based on analysis. Assertion, reasoning questions are based on analysis. And then comes a very important evaluation. My dear friends, evaluation questions develop decision-making skill among learners. In evaluation questions, the children need to take a decision. The children need to justify their decision also. The questions where decision is required. Which one is best? Which one is better? Which would you select? So first, what do? You compare the two concepts, and on the basis of comparison, we take a decision. So evaluation thinking skill develops decision-making skill among children, and then becomes the highest level of cognitive domain. That's a creation. So what are creation-based questions? We'll discuss in the later on also. The creation-based questions are the questions where you need to use two or more than two concepts. The children need to integrate two or more than two concepts to generate a new concept. In these questions, the children will be given an unknown situation where they need to apply more than two concepts to solve the problem. So when we plan a lesson, my dear friends, make it sure that your learning objectives, your learning outcomes, all assessments are aligned with Bloom's taxonomy. And these are the suggestive learning outcomes as per Bloom's taxonomy. These are not the ultimate. These may change from teacher to teacher, from topic to topic. So these learning outcomes define the questions which are based on their remembering skill, understanding skill, applying skill, analysis skill, evaluation and creation skill. So it is not mandatory to define the learning outcomes which can touch all the 6 levels of cognitive domain. You can touch 3, you can touch 4. But at least try to take one learning outcome from the higher order thinking skill. Maybe analysis, maybe evaluation. And if it is possible in a particular topic, you can take the learning outcomes, you can plan the learning outcomes, you can decide the learning outcomes based on all 6 levels of cognitive domain. Now let's begin with today's topic, that is the magnetic field due to current carrying conductors. The previous knowledge required to understand this topic is what is magnetic field? What are magnetic lines? And before coming to this topic, the children must have learned what is magnetic field, what are magnetic lines due to a bar magnet. A bar magnet is a permanent magnet. And what is the magnetic field? Magnetic field is a space around a magnet, the region around a magnet where its effect can be experienced. And what are magnetic lines? Magnetic lines are nothing but imaginary pictorial representation of magnetic field. It's a pictorial representation of magnetic field around a magnet. And in case of a bar magnet, if you can see the figure here, this is an animated diagram of the magnetic lines and magnetic field around a bar magnet. And you can see in that diagram that the magnetic lines are directed from north to south pole outside the magnet. And within the bar magnet, these magnetic lines are directed from south to north. So it is very clear, it is crystal clear that the magnetic lines make closed loops. So it must be clear to the children that the magnetic lines make closed loops. And at any point, if we want to find out the direction of magnetic field, there are two options. In first option, we can place a magnetic needle there. You can see this magnetic needle here in my hand. So you can place the magnetic needle there. And the direction in which the north pole of the magnetic needle would point, that would be the direction of magnetic field. And the second point is that we can draw a tangent at that particular point. The direction in which the tangent is aligned, that would be the direction of magnetic field at the given point. No two magnetic lines can ever intersect. This must be clear to the children. And the magnetic monopoles do not exist. The isolated magnetic poles do not exist. The north is always associated with the south pole. So this is the previous knowledge which is expected to be there before beginning with the topic. My dear friends, Orested was the scientist who laid the foundation that a current carrying conductor behaves as a magnet. Before 18th century, the electricity and magnetism were used to be the different phenomena. Even though some sailors, while traveling, had found that whenever the lightning strikes their ship, the magnetic compass in their ship gets deflected in the different direction. It means before 18th century also, some of the sailors have noticed this thing, that atmospheric electricity has ability to deflect the magnetic needle. But they did not notice it. So before 18th century, it was believed that electrical forces and magnetic forces are distinct forces. They are different forces. In July 1820, Danish scientist Christian Ørsted accidentally discovered a relationship between electricity and magnetism while demonstrating the heating effects of electric current in a classroom. Ørsted noticed that when he turned on an electric current by connecting a cell with the wire, the compass needle held nearby deflected away from the magnetic node where it normally pointed. So Ørsted accidentally found that a magnetic needle placed close to the current carrying conductor is able to give deflection. If the magnetic needle is giving the deflection, it is crystal clear there is a magnetic field around it. And there was no source of magnetic field around that magnetic needle. Hence, the Ørsted concluded that the source of magnetic field is the current carrying conductor that was placed very, very close to the magnetic needle. Although that deflection was very weak, but the Ørsted knew that something significant has happened. It means the world came to know about the new branch of physics, and that is called as electromagnetism. So there is a crystal clear relationship between electricity and magnetism. So you can see in the diagram to the extreme left side, the magnetic needle is placed below the current carrying conductor. And the north pole of the magnetic needle is giving the deflection towards east. And in the middle diagram, you see the same magnetic needle is now placed above the current carrying conductor. And now the north pole of the needle is giving the deflection towards west. So this is clear now that above the current carrying conductor and below the current carrying conductor, the direction of magnetic fields are different. They are opposite to each other. And to the extreme right, you can see the diagram. It's an animated diagram. You see one compass needle is above the current carrying conductor and one compass needle is placed below the current carrying conductor. So when the current is passed to the conductor, both the needles are giving the deflection in opposite direction. The red part is showing the north pole, the black part is showing the south pole. You see their red parts are getting deflected in the opposite directions. So magnetic field is there above as well as below the conductor. But the direction of magnetic field is 180-degree opposite to each other. The direction of deflection of magnetic needle can also be found by using Ampere's swimming rule. So we can tell our children that they can apply the Ampere's swimming rule to find the direction of deflection of magnetic needle. The rule says that if a man is swimming in the direction of current, means his head is pointing in the direction of current, then the north pole of the magnetic needle placed above the current carrying conductor would be deflected towards his left hand. But if the same man is swimming below the current carrying conductor, then the north pole would be deflected towards his right hand. And this rule can also be remembered as the term snow. And what does the snow mean? The snow means that if the current is flowing from south to north of the magnetic needle, then the north of the magnetic needle would be deflected towards west. So if the current is from south to north, then the north of the magnetic needle would be deflected towards west. So with the help of Ampere's swimming rule or the snow rule, the children will be able to find out the direction of deflection of magnetic needle when it is placed above or below the current carrying conductor. Now it's a time to think, my dear friends, does magnetic field produced by a current carrying conductor depend on the shape of the conductor? I need your responses. What do you think? Does the magnetic field produced by a current carrying conductor depend on the shape of the conductor? So you can see in my hand, there are three types of conductors, if you could see. It's a straight conductor. It's an insulated copper wire. It's a straight conductor. It's a circular coil. You can see it's a circular coil having few turns. The circular concentric turns. It's also a conductor. And this is a helical coil, also called as solenoid. So I have three different types of conductors: straight, circular, and helical. What do you think? In all three cases, we will have same magnetic field or different magnetic field. So when I say magnetic field, it means both magnitude as well as direction. I need your responses, my dear friends. If they are physics teachers today, please respond to this question in yes or no.
Yes, sir, it will be having different magnetic fields, different shaped magnetic fields.
So yes, good. So it is clear after looking at this slide that the magnetic field produced by a current carrying conductor does depend on its shape. In the first diagram, you can see, in the first figure, you can see we have a straight conductor. It's a current carrying conductor. And in the second case, we have a circular coil. In the third case, you can see we have a helical coil called as a solenoid. In all these three cases, you can see the pattern, the strength, the direction of the magnetic field is different. So it is very clear to us now that the magnetic field produced by a current carrying conductor depends on the shape of the conductor also. So first of all, we will discuss the magnetic field due to an infinitely long current carrying straight conductor. My dear friends, before I proceed with this topic, I just want to discuss with you one point, and that is called as inclusive education. This is very important point, inclusive education. It's a very broader term, but I will discuss with you only one aspect of the inclusive education, and that is differentiated instructions. See, in a classroom, we have slow learners, we have gifted learners. So it is the responsibility of the teacher, it's the responsibility of the mentor to cater the needs of both type of learners, slow learners as well as gifted learners. The gifted learner in a classroom are those children who are highly focused, logical, and they normally participate in the various type of competitive exams. Like NTSC, we used to have NTSC, and a various type of Olympiads. So it means for gifted children, we cannot limit our learning objectives to the NCERT textbook only. We need to go beyond the NCERT. So in today's session also, I will be discussing with you some additional informations related with the current topic. So I hope you won't mind it and you won't put the message in the chat box that this is not the part of the syllabus, this is the deleted syllabus. Because the gifted students need some more informations apart from the NCERT text. So today I have included the additional informations also, which you can provide to the gifted children. So let's come to the session again. The magnetic field due to an infinitely long current carrying straight conductor. After Oersted experiment, it was concluded that a current carrying conductor acts as a source of magnetic field. It was also found later on that magnetic field due to current carrying conductor depends on the various factors, including the shape of the conductor. So let us consider a straight conductor in the form of an insulated copper wire. And for experiential learning, we can pass this straight conductor to the center of a thick cardboard sheet. And the two ends of the wire are connected with the cell with the help of a rheostat and a key. The rheostat is a variable resistance. It can be used in the circuit to change the magnitude of the current. So when we switch on the circuit, the current starts flowing through the straight conductor. And we place a magnetic needle very close to it, we'll find that the magnetic needle is giving the deflection, and deflection in the magnetic needle shows the presence of magnetic field around the current carrying straight conductor. If we sprinkle iron filings randomly on that thick cardboard sheet, and we tap the cardboard sheet gently, we will find that the iron filings arrange themselves in a specific pattern. And if you look at the diagram there, that specific pattern is concentric circles. and the center of all circles lie on the wire. So it means the magnetic lines around a straight conductor are in the form of concentric circles, where the center of all circles lie on the wire. It's time to think again. I need your responses. Please give your responses. What do you mean by infinitely long straight conductor? What is an infinitely long straight conductor? If I say this few centimeter long conductor is infinitely long, then on what basis I'm saying this is infinitely long, although it has a finite length? So this point must be clear to the children in the classroom teaching. Why, on what basis I'm saying this is infinitely long? And why there is need to tap the cardboard sheet after sprinkling the iron dust on it? Why do we tap it? Why the instructions are there in the experiment also that we need to tap the cardboard sheet? I need your responses, my dear friends, physics teachers. Please give your response.
Iron fillings will adjust themselves according to the magnetic field of the straight long conductor.
See, the answer to first question is the infinitely long straight conductor is that conductor whose length is much larger as compared to the perpendicular distance of the observation point from the straight conductor. It means the point where we are calculating the magnetic field is lying very, very close to the straight conductor. It means the L, that is the length of the conductor, is much greater as compared to small r. Here, small r is representing the perpendicular distance from the current carrying conductor. So in that sense, the conductor is infinitely long, even though it has a finite length. The answer to second question is why there is a need to tap the cardboard sheet. We tap the cardboard sheet due to two concepts. First, to overcome the inertia. We will have to transform the inertia of rest into inertia of motion. And second is to overcome the force of friction. Unless or until you overcome the inertia and the force of friction, the iron filings won't move under the effect of magnetic field. So these two basic informations must be conveyed to the children in the classroom teaching while discussing this topic. Now, this slide represents how we can find out the direction of magnetic field at a particular point around a straight conductor. At any point where you want to find out the direction of magnetic field, place the magnetic needle. You can see in the animated diagrams here. So when you place the magnetic needle at a particular point, the magnetic needle gives the deflection. The direction in which the north pole is pointing, the direction in which the north pole of the magnetic needle is getting deflected, is the direction of magnetic field at that particular point. So you can see in both the diagrams, when the current is passed through the conductor, all magnetic needles placed around it are getting deflected. And you see the pattern in which they all are getting deflected. अगर मैं सभी points को mark करके join करूं, I would get a circle. So this again proves that the magnetic lines around a current carrying conductor are in the form of concentric circles. Now, we have seen this thing that the magnetic lines around a current carrying conductor are in the form of concentric circles. Now, the next issue is, are these circles clockwise or anti-clockwise? And to find the direction of magnetic lines around a current carrying conductor, whether they are clockwise or anti-clockwise, we can use right hand thumb rule, which you can see in the diagram here. The right hand thumb rule says that if we hold the current carrying conductor in our right hand in such a way that the thumb is pointing in the direction of current, then the direction in which the fingers of the right hand are getting curled will be the direction of magnetic lines. So it is crystal clear from this diagram, from this rule, that if the current is vertically upwards, then the magnetic lines would move in the anti-clockwise direction. But if you see, if the thumb is vertically downwards, means the current flowing through the conductor is vertically downwards, then you see the direction in which the fingers are getting curled. This is a clockwise direction. And when the thumb is pointing vertically upwards in the direction of current, you see the direction in which the fingers are getting curled. This is the anti-clockwise direction. So this is a very simple rule given in NCERT textbook, which the children can apply to find out the direction of magnetic lines around the current carrying straight conductor. There is another rule that's called as the Maxwell's corkscrew rule. And that has the same application. This is also used to find out the direction of magnetic lines around a current carrying straight conductor. And the rule says that if we rotate the screw in such a way that its tip is pointing in the direction of current, then the direction of rotation would be the direction of magnetic lines. So it means if the screw is rotated anti-clockwise, my dear friends, if the screw is rotated anti-clockwise, then its tip always points in a vertically upward direction. When the same screw is rotated clockwise, its tip points in a vertically downward direction. So you can show a screw in the classroom, right-handed screw, and keeping the screw in the right hand, try to rotate it in the front of the class, clockwise or anti-clockwise direction, and see in which direction its tip is pointing. So that would also, this rule would also help the children to find out the direction of magnetic lines around a current carrying straight conductor. Now I have used an ICT pedagogy here to represent the magnetic field pattern around a current carrying straight conductor. In the first diagram, you see the current is flowing in a vertically upward direction. And the magnetic lines around this current carrying conductor are in anti-clockwise direction. In the second diagram, again, you see when the current is switched on, you look at the deflection of the magnetic needles. Look at the pattern of the deflection of magnetic needles kept around the current carrying conductors. That pattern is resembling like a circle. And then you see in the middle diagram, when the current is flowing through the conductor, the magnetic needle kept above and below the current carrying conductor are showing the deflection in opposite direction. And fourth and fifth, again you see the current is flowing, and look at the pattern of the magnetic lines. They are in the form of circles. So we can show this picture in the classroom with the help of smart boards. We can share this ICT pedagogy with the class through the class groups. The children will be able to understand. They will be able to analyze the concept of the current carrying conductor, how they produce the magnetic field around it, what is the pattern, what is the direction. So there are so many factors which affect the magnetic field due to a current carrying straight conductor. If we increase the strength of the current flowing through the conductor, You see the magnetic compass placed near the conductor gives a very strong deflection. If the strong deflection is appearing in the magnetic needle with the increase in the strength of the current, this clearly shows that the magnetic field is directly proportional to the strength of the conductor, the strength of the current flowing through the conductor. So as we move away from the current carrying conductor, the deflection in the magnetic needle becomes poorer and poorer. The magnetic needle gives a very weak deflection when it is kept far away from the current carrying conductor. So this clearly shows the magnetic field is inversely proportional to the distance from the conductor. And my dear friends, this distance should be taken perpendicularly. So it is clear that magnetic field of a current carrying conductor depends on the two factors. One is the current flowing through the conductor. Another is the perpendicular distance from the current carrying conductor. And if we remove the proportionality, you look at the slide. If we remove the proportionality, we get a constant here, which is mu naught by 4 pi. Now, this is an additional information which you can provide to the gifted children in your classroom. That the constant here is mu naught by 4 pi. Now, what is mu naught? This mu naught is called as permeability of the free space. Now, what is permeability? It is the ability of a medium. It is the ability of a substance to allow the magnetic field to pass through it. Permeability represents the extent up to which a substance can be magnetized. And the value of this mu naught is 4 pi into 10 raised to power minus 7. You can give this information to the students who are gifted, who are focused, and who need the additional information. And the next conductor is a circular coil, a current carrying circular coil. My dear friends, every elementary part of a current carrying circular coil produces a magnetic field. And the magnetic field produced by every elementary part of a circular coil gets added up at the center. So the magnetic field that we actually get at the center is the sum of magnetic fields produced by every elementary part of the current carrying conductor. We can show the similar activity. We can perform the similar activity as we did in the straight conductor. We can take a straight conductor, we can fold it into a circular loop whose two ends are passing from the two holes made on a thick cardboard sheet. And if we join the two ends of that circular wire with a cell with the help of a rheostat and a key, and we place a magnetic needle close to the arms of the circular wire, what do we observe? That the magnetic needle gives the deflection. So that shows the presence of magnetic field around a current carrying circular coil. So you can see these are beautiful diagrams representing the pattern of magnetic lines around a current carrying circular coil. In the first diagram, you see when the current is passed through the circular coil has few turns. See how the magnetic lines are formed and how the iron dust is getting arranged. You look at the first diagram. The iron dust present on the cardboard is getting arranged in the form of concentric circles. And look at one more thing. These circles are becoming larger and larger as we move away from the current carrying portion of the circular coil. So this clearly shows that in case of a circular loop also, the magnetic lines are in the form of concentric circles. And as we are moving away and away from the part of the current carrying coil, the circles are becoming bigger and bigger. Look at the center. in the second diagram or the third diagram, look at the center of the current carrying coil. The magnetic lines are almost parallel to each other. So that clearly shows that in the middle, at the center of the current carrying coil, the magnetic field is uniform. My dear friends, the parallel lines, make it clear to the children in the classroom, the parallel magnetic lines represent a uniform magnetic field. Now, how can we find out the direction of magnetic field at the center of a current carrying circular coil? To find the direction of magnetic field at the center of a current carrying circular coil, we have right hand rule. So what does this right hand rule say? The rule says that if we curl the fingers of the right hand in the direction of current flowing in the circular coil, then the direction in which the thumb of the right hand would point would be the direction of magnetic field. I again repeat, we are to hold the circular coil in such a way that the fingers of the right hand are pointing in the direction of current. And you see, if the current is anti-clockwise, then the thumb is pointing in the perpendicularly outward direction. And if the current flowing in the coil is clockwise, the thumb is pointing in perpendicularly downward direction. So it means the direction of magnetic field in case of a current carrying circular coil is perpendicular to the plane of the coil. This is very important information which needs to be told to the student in the classroom, that the direction of magnetic field in a current carrying circular coil is perpendicular to the plane of the circular coil. And one very important point here, as I told in the beginning of this session, the isolated poles do not exist. The magnetic monopoles do not exist. My dear friends, when the current flows through a circular coil, the circular coil behaves as a complete magnet. So when we say complete magnet, it means its one face is behaving as a north pole and other face is behaving as a south pole. Now, how can we find out that which face is acting as a north pole and which face is acting as a south pole? And for that, we have a clock rule. And that is called as a clock face rule. And according to this rule, if we stand in front of a current carrying coil and we look at the direction of current, if the direction of current is anti-clockwise, look at the diagram here. If the direction of current is anti-clockwise, then that face of the circular coil would behave as a north pole. And if the direction of current in that particular face is clockwise, that face would behave as a south pole. So looking from one face, if the current is anti-clockwise, that face would be north pole. And looking from the opposite face, the direction of current would be clockwise and that would behave as a south pole. So current carrying circular loop is a complete magnet. It's a complete dipole having north and south polarity. My dear friends, let us watch this video. This is a beautiful video that would explain to you that how a current carrying circular coil behaves as a magnetic dipole, how it produces the magnetic field, how can we find the direction of that magnetic field, and what is the pattern of magnetic lines around a current carrying circular coil. This is a beautiful video of about 6 minutes. Please focus on it.
Magnetic field due to a current carrying circular coil. We have already studied that when current is passed through a straight conductor, a magnetic field is produced around the conductor. This region of magnetic field can be represented as concentric circles around the wire. So we can say that a cylindrical shell of magnetic lines of force surrounds the entire conductor. The direction of these lines of force can be found by the right-hand thumb rule or Maxwell's corkscrew rule. Both these rules can be verified with a compass needle also. Now, modify the straight conductor to form a circular loop and pass it through the two holes in a cardboard. Connect the ends of the coil and a rheostat in series. Sprinkle some iron filings randomly on the cardboard. Switch on the key of the circuit and adjust the rheostat to allow a current of about 2 amperes to pass through the conductor. It can be seen that the directions of the current flowing through each arm of the circular loop are mutually opposite. Now, as we gently tap the cardboard, we can see that the iron filings align themselves in a specific pattern. They are in the form of circles around each arm of the conductor. These circles prove the presence of a magnetic field. To find the direction of these magnetic lines of force, we apply the right-hand thumb rule. According to this rule, we can find that the direction of the magnetic lines of force around the arm of the conductor with upward flow of current is anti-clockwise, while that around the arm of the conductor with downward flow of current is clockwise. In other words, as the direction of the current in each arm of the conductor is mutually opposite, the direction of the magnetic lines of force is also opposite to each other. Conversely, within the space enclosed by the coil, the lines of force are in the same direction. Let us study the various properties of the magnetic field produced due to the current-carrying circular coil. Observe that the lines of force are circular near the arms of the conductor. But as we move from the arms towards the center of the coil, the lines of force become almost parallel to each other. Thus, near the center of the coil, the magnetic field may be considered to be uniform. Hence, it can be said that as the distance from the center of the conductor increases, the magnetic field intensity decreases. At the center of the coil, the magnetic field is maximum and is directed perpendicular to the plane of the loop carrying current along its axis. Next, increase the current flowing through the conductor from 2 to about 6 amperes and tap the board gently. We can see that more iron filings get aligned with the magnetic field lines. That is, the number of magnetic lines of force increases as the magnetic intensity increases. Now, increase the radius of the coil. You can see that the magnetic field intensity decreases. The magnetic field produced around the coil proves that the coil behaves like a magnet and has a north and south polarity. The clock rule is used to find the polarity of a current-carrying coil. If we look at this particular face of the coil, we can see that the current is flowing in an anti-clockwise direction. Moreover, the magnetic lines of force emerge out of this face of the coil. Hence, this end is said to be the north pole. Similarly, looking from the other side of the coil, the current is found to be flowing in the clockwise direction. The magnetic lines of force enter into this face of the coil. Therefore, this face is considered to be the south pole. Hence, the clock rule states that when looking at one particular face of the coil, if a current is found to be flowing in an anti-clockwise direction, that face of the coil is considered to be the north pole. If the current flows in the clockwise direction, that face of the coil is considered to be the south pole. Summary. magnetic field is produced around a circular coil carrying current. The direction of these magnetic lines of force can be found using the right hand thumb.
So, my dear friends, I hope you have liked this video, and that made the concept of magnetic field around a current carrying coil very clear. So it is very clear from this video that the magnetic field produced by a current carrying conductor in the shape of a circular coil depends on the two factors. One is the current flowing through the conductor, circular conductor. Another is the radius of that circular loop. So if we combine these two, we get the magnetic field at the center of a current carrying circular conductor is directly proportional to I upon R. And if the children ask from you that what will happen if we remove the proportionality and gets a constant here, what will be the value of that constant? So you can provide an information to your students in the class that that constant is actually here in this particular case mu naught by 2, where mu naught is again the permeability of the medium. To what extent the medium can be magnetized, that is the meaning of permeability. And the value is 4 pi into 10 raised to power minus 7. If the circular coil has n turns, then every turn will produce an equivalent magnetic field at the center in same magnitude as well as in same direction. So therefore, the net magnetic field at the center would be the sum of magnetic fields produced by all the n turns. And as a result, the net magnetic field at the center becomes mu naught Ni upon 2R. And next we have the magnetic field due to a current carrying long solenoid. So what is a solenoid? A solenoid consists of an insulating long metallic wire closely wrapped in the form of a helix. A long solenoid is that whose length is very, very large as compared to its radius. When a current is passed through the solenoid, then each turn of the solenoid can be regarded as a circular loop. You can see here, my dear friend, this is a solenoid. This is a helical coil. See, it consists of a very large number of circular turns. Every circular turn will have its independent magnetic field. And at any point, if you find the magnetic field, that would be the sum of magnetic fields produced by all the circular turns present in this helical coil. Every single circular turn will have its own magnetic field. If there are 10,000 turns, let us suppose, then there will be 10,000 magnetic fields at one particular point. And the net magnetic field would be the sum of all the magnetic fields. My dear friends, let us watch a very short video to get an idea of the magnetic field lines pattern around a current carrying helical coil or a solenoid. It's a very short video that will describe the pattern of magnetic lines around a current carrying helical. See how the iron dust is getting arranged. The field around a current carrying solenoid looks similar to that of a bar magnet. So see the beautiful diagrams here representing the magnetic field due to a current carrying helical coil. or a solenoid? It's a very important topic in 10th standard science. As I told you that at any point, the net magnetic field is the sum of magnetic fields produced by all the circular turns present in the solenoid. It has been observed that outside the solenoid, these magnetic fields cancel each other. These magnetic fields oppose each other. As a result, outside the current carrying solenoid, the magnetic field is very, very weak. And for practical purpose, we take it as zero. But inside, in the interior of a current carrying solenoid, all these magnetic fields add up. As a result, we get a very, very strong and a uniform magnetic field inside the current carrying solenoid. If you look at the diagram here, these are animated diagrams. If you look at the first diagram, you will find that inside the current carrying solenoid, the magnetic lines are almost parallel to each other. The magnetic lines is a very important question in 10th standard physics. If the magnetic lines are parallel in the interior of a current carrying solenoid, it represents that the magnetic field there is uniform. So in the interior, in the middle of a current carrying solenoid, there is a strong magnetic field which is almost uniform. And one very peculiar property here, that this magnetic field resembles the magnetic field due to a bar magnet. Now, the many students may ask this question, that why the magnetic field of a bar magnet is similar, is equivalent to the magnetic field of a current carrying helical coil. My dear friend, there is a beautiful video. Watch this video and you will come to know that why and how a current carrying solenoid is equivalent or similar to a bar magnet. This will make the concept clear. It's a beautiful video, my dear friends. Look at this.
Saw that if you have an electric current carrying loop, then it generates a magnetic field that's very similar to that of a bar magnet. But it doesn't quite resemble a bar magnet, because if you look inside the bar magnet over here, then you can see that there is this long region where the magnetic field is pretty straight. It's not exactly straight, but pretty much straight and parallel to each other. Can you see that over here? However, if you look at our artificial magnet over here, then notice that this region where the magnetic field lines are straight is very short. And so this resembles not a big bar magnet, but a very tiny bar magnet. So now the question is, how do we create something that generates a magnetic field perfectly like a large bar magnet, where we have a large region where the magnetic field lines are pretty straight inside? That's what we should find out now. So how do we do this? Well, what if we bring in another current carrying coil? Another current carrying coil and place it down here somewhere. Then it's identical to this one, so it's going to produce an identical magnetic field of its own. And I'm going to show that in a different color over here. But remember, so you know what's going to happen? The two magnetic fields are going to merge together and give us a new magnetic field that's going to look somewhat like this. Ooh, look at the combined magnetic field. We now have a straight region over here. We also have a straight region over here. But in between, it's kind of bulky. So how do we straighten this out? Well, I'm pretty sure you can guess now. Let's add one more current carrying coil in between over here, and let's see if it straightens out. So if we introduce another current carrying coil, again, it's going to generate its own magnetic field, which I have shown in another color. And I'm pretty sure it's looking like a mess right now. But again, the magnetic fields don't ever intersect. So now this magnetic field will merge with the previous magnetic field. And together, the magnetic field of all the three coils together is going to look somewhat like this. Hey, look at this magnetic field. We're getting close to what we want. But again, we have these little bulging out regions. We have little wings over here. And I'm pretty sure you know what to do next. Well, just keep adding more and more coils in between. So you know what? Let's go crazy. Let's say we add 50 coils in between. I'm not going to show all the 50 coils. I'm just going to show only a few one. But let's imagine we add something like 50 coils. I'm pretty sure you can guess what's going to happen. The magnetic field inside is going to become almost straight now. And these regions are also going to straighten out. And so the new magnetic field of all these coils together will look like this. And ta-da! We have done it. This looks exactly like what we wanted. So if we have lots and lots of current carrying coils, then it resembles a bar magnet. Now, of course, to make this construction easier, instead of taking different loops and keeping it on top of each other, we could take a single wire and then coil it like a spring to give us multiple loops. Somewhat like this. See, we've taken a wire, you coil it multiple times and pass current through it. And since we're getting a magnet due to electric current, we call such magnets electromagnets. And this particular electromagnet, which has a shape like a pipe or a tube because of so many coils, we give it a name. It's called a solenoid. And the word comes from German, Solen, which means pipe, and oidus, which means shape. So whenever you coil something so much that it looks or resembles like a tube or a pipe, we tend to call it a solenoid.
So I hope you have liked this video. And now we will be able, after watching this video, we will be able to answer to our children that why a current carrying solenoid is equivalent to a bar magnet in terms of the magnetic field pattern. The magnetic field produced by a current carrying solenoid at a point in the interior, not outside. I told you that outside magnetic field is very, very weak. At a point in the middle is directly proportional to the current flowing through the solenoid and the number of turns per unit length. So when we say number of turns per unit length, it means the number of turns present in a unit length, means 1 meter length of the solenoid. So the number of turns per unit length can be obtained by dividing the total number of turns with the length of the solenoid. So the expression for the magnetic field becomes that B is directly proportional to Ni upon L. And again, if we remove the proportionality, we get a constant here, and that constant is mu naught. So the magnetic field intensity, the magnetic field strength present at any point in the interior of a current carrying solenoid is given as mu naught Ni upon L. My dear friends, the NEP 2020 calls for a shift from an assessment system that is summative and which is basically test the rote memorization skills to one that is more regular and formative, that is more competency based, promotes learning development for our students, and test higher order skills such as analysis, critical thinking, and conceptual clarity. It means we need to plan such assessments after every lesson, which can develop higher order thinking skills among our children. The children must become critical thinker. They must become creative. They must become resilient. They must have decision-making skill, problem-solving skill. So assessment questions should not be based merely on rote memorization. And this is an assessment as planned by me for today's session, which is based on Bloom's taxonomy. And there are the questions which are based on remembering skill. There are the questions which can test their understanding skill. There are the questions you see which test their applying skill, analysis skill, evaluation, and creation. So we, the mentors, we, the teachers, have a responsibility to develop low order thinking skill as well as higher order thinking skills among our children. And this is the only way we can justify the vision of NEP. Thank you. So if you have any question, any query, you are most welcome.
My dear friends, do ask me questions. If you have any query.
Okay, thank you very much.
My sincerest regards to
our mentor, Dr. A.K. Sharma, sir.
Thank you very much. Today's session was wonderful. First session was taken by Dr. Gurpreet Kaur. It was very nice, although I belong to the subject, but I have thoroughly enjoyed. There are many things which are beautifully explained, and
Thank you, sir.
it is the way the teacher should strike the chord, because many concepts are there which are not there in the NCERT, as Mr. Hartendra also said. So what is the characteristic feature of a good teacher is that we should not confine to the textbook. We should go beyond the textbook, make the version simplified for the students and give them so that they should know. And Dr. Gurpreet Kaur has well explained the mystery of the life. Beautiful presentation.
Thank you, sir.
The chapter was also very good in production. And the second session was Mr. Hartinder. I was looking to the chat box. Some of them was saying that it is a very good session, very descriptive, very elaborative. And he has explained various concepts related to this magnetic field. And it was supported by the videos and by the diagrams. So I think that the concepts should be very clear to the teachers. Sometimes when we teach in the class, some concepts are missing, some links are missing. So in case any such problem you are facing, it is the right platform to discuss all these things, not only about this chapter. If you have any problem about any other chapter, you can freely talk to us at the last. So thank you very much, Mr. Hartinder Sharma and Dr. Gurpreet Kaur.
Thank you, sir. Thank you so much.
You are superb. Thank you.
Thank you, sir.
And
Yes, sir.
now it's your turn.
Thank you, sir. Thank you so much, sir. So my sincere thanks to the resource persons. It was a lovely session, very well explained and informative. So we should end the session today, conclude with the Shanti Mantra.
ॐ पूर्णमदः पूर्णमिदं पूर्णात् पूर्णमुदच्यते पूर्णस्य पूर्णमादाय पूर्णमेवावशिष्यते ॐ शान्ति शान्ति शान्तिः हरिः ॐ श्रीगुरुभ्यो नमः हरिः ॐ
Thank you so much. So can we just leave the session? So I request the teachers to please leave the session.